Hello everyone, my name is Malay Basu. I'm a faculty at University of Alabama at Birmingham. Today I'll be talking about our work on measuring proteome complexity in cancer using linguistic modeling. Proteome imbalance have been implicated in various disease and abnormal state of cells. Many cellular processes can contribute to proteome imbalance, such as alternative splicing, abnormal translation, etc. Drugs such as carfilzomib and bortezomib have been used to target proteome imbalance. In cancer, gene copy number and structural alteration leads to proteome imbalance. One of the models of cancer evolution, called Atovistic model, proposes that cancer resolves from the breakdown of multicellularity to a retrogression into a unicellular state where cell division takes priority over multicellular cooperation. We decided to investigate this using a linguistic modeling technique that we published recently. Ever since the human genome sequence, the phrases book of life or the language of genes have been used metaphorically. In our work, we use these phrases not metaphorically but literally. Genomes can be thought of as a book or corpus. Genes or protein sequences can be thought of as sentences, and protein domains can be thought of as words. How similar are words and domains? David Sells in a 2002 paper showed that the distribution of domain in each genome is very similar to distribution of words in Shakespeare's Romeo and Juliet. Such distributions follow Zipf's law, a kind of power law, which is typically identified as a straight line in a log-log plot. If we plot the frequencies of domain with their ranks in human genome, we find that the slope of the curve is minus 0.99 or close to 1, which is almost identical to the value minus 1 reported for natural languages. A very popular technique in linguistics is Engram Analysis, where we take consecutive words in sentences to find their meaningful association. The simplest form of Engram Analysis is bigram, where two words are taken at a time. Consider a genome containing only two proteins, protein 1 and protein 2. Protein 1 has the domain architecture of A, and B, and protein 2 has the domain architecture of C, A, and D. So the bigrams from protein 1 will be AA and AB, and the bigrams from protein 2 will be CA and AD. I have already shown you before that a single domain distribution follows a similar power law as natural languages. Here I am showing that even the domain bigrams follow power law. Here we see 4 representative species, with human having a value of minus 1.86, which is very similar to the value minus 2 reported for natural languages. So far we have seen that the frequency distribution of single word or domains, which is also called unigram, have similar values in natural languages and genomes. Even the bigram frequencies also have similar power law characteristics. Given an unknown language, the most simplest analysis that can be done is Engram language modeling. Engram language model calculates the probability of a word given a set of preceding words. Depending on the number of the preceding words, called order, they are called unigram for 0 preceding word, bigram for 1 preceding word, trigram for 2 preceding words, etc. The Engram models are also generative. Given Engram model from a genome like human, we can generate a random human genome. To give an intuitive feeling of language modeling, Let's say you are typing a word in Google search. The browser suggests alternatives based on the language model according to the probabilistic association of words. To understand language model, we also have to understand the concept of entropy as it is applied in language modeling. Like in thermodynamics, randomness of a system is measured as entropy. Relative entropy of a language model is the amount of information gained going from the random arrangement of words to meaningful arrangement of word ordering, which is dictated by grammar. There are around 7,000 languages in the world divided into 19 linguistic families. It turns out that all human languages have relative entropy of 3.6 bits per word. The reason behind this universal relative entropy of all human languages is not really known, but it has been extensively used to determine whether a series of symbols represent a language. The technique has been extensively used to identify whether the plaques discovered from the Indus Valley civilization represent a language or not. Formally, a bigram model is the conditional probability of a domain Dn given its preceding word Dn-1. This can be calculated by dividing the count of the domain pair Dn-1 Dn by the count of the first domain in the genome. The problem with Engram modeling is that the models are very sparse and it requires some kind of smoothing before they can be used. Over the years, many different kinds of smoothing techniques have been developed. So we created domain language models from 450 eukaryotes, 180 archaea, and 4,000 prokaryotes. Unigram entropy is simply the sum of weighted log values of frequencies of domains in the genome. This can be considered a randomized version of the genome, as if all the domains in the genome are put in a bag of words and shaken. Bigram entropy is calculated similarly as the sum of the weighted conditional log probabilities. And the relative entropy, or the information gain, can be calculated by subtracting bigram entropy from the unigram entropy. When we looked at the relative entropies in all the genomes, we found that except in metazoa and a small group of archaea, the relative entropy lies in a close range around 1.2 bits. The phenomenon is very similar to the universal relative entropy as seen in case of natural languages. Let's see how these grammars evolved. For this, we need to know about cross entropy, or perplexity is the measure of uncertainty of a genome given a language model from another genome. So we modify the bigram equation. The cross entropy of a target genome G given the model M is calculated by using the counts of bigram from the target genomes, but the conditional probabilities come from the model. Self entropy is the cross entropy where model and the target genomes are the same. We can normalize the cross entropy values by the self entropy and convert the values into distance. Evolutionary distant genomes will have greater distances and similar genomes will have smaller distances. This results in a distance matrix which can be used to create a phylogenetic tree of the bigram models. We created the tree for 38 eukaryotic genomes. The tree shows remarkable similarity to existing tree topology. Depending on the root placement, the tree conforms to both the crown group and unicorn bicon topology, the two prominent evolutionary models of eukaryotes. Now, given this background, to measure proteome complexity in tissues, we decided to use gene expression data as a weight in the bigram model. The equation is very similar as we discussed earlier, but the weights in the equation comes from the expression data. This is the equation for the Unigram model, and this is the equation for the bigram model. First, we use the expression weighted data to measure the proteome complexity in human tissue using GTEx data. On the left side, you see the relationship tree which perfectly clusters related tissues together. On the right hand side, we see the relative entropy of the tissues. The values lie from 3.16 in spleen to 4.28 in liver. The average of this value is around 3.7. The relative entropy of the human genome, shown as the gray line, is around 2.2 bits per domain. So, when it comes to human tissue, the liver is the most complex of all the tissues. Next, we looked at what happens in cancer. We used upper quartile normalized data to create expression weighted language models from all TCGA cancer types. We assume that the RNA expression is a representation of the proteins in the cells, which is a reasonable assumption, at least in steady state. When we measure the proteome complexity in cancer, we find that the median value of complexity varies amongst cancer types. The dotted line here represents the average complexity of human tissue, while some cancers like LAML and OV, the relative entropy is relaxed. Median complexity in cancer types rises lower than the tissue complexity. This also means that the gene expression in these cancer types are more random from the human tissue and might follow the atavistic model of cancer evolution. The interesting observation is that the deadly cancer types with poor prognosis, like mesothelioma, liver carcinoma, pancreatic cancers, all have higher relative entropies compared to human tissue. It means that these cancer types are driven by streamlining, presumably due to adaptive evolution. The relationship is more evidently shown here if we classify all samples in TCGA into two categories, one with higher relative entropies compared to the average relative entropy of human tissue, and a second with lower relative entropies. We find that the higher relative entropy is associated with higher mortality rate, shown by the brown line here. So, in conclusion, we have shown that atavistic model may hold true for only certain cancer types, but not all. Higher protein complexity or adaptive evolution is associated with higher mortality. We predict that drugs targeting proteome imbalance might work for atavistic cancer types, but not for adaptive cancers. Many people have contributed to the work that I presented here. Particularly important is Eugene Koenig at NCBI. Ligia did bulk of the work presented here. Questions or Rotten Tomatoes?